thanks very much, and I'll try and keep it brief. When I was told I had 10 minutes to talk to you about product development and project management, I thought, oh, I could talk for days, but I won't. I've called my talk The Science and Fiction of Drug Development, partly because I really like science fiction books, but also I understand that most of you have some concept of the pathway of taking a product from preclinical to phase 4, but not all the compounds are the same, and even though the pathway, there are goals that need to be met, you've got to tailor it every time for what your compound is telling you. There's no point trying to push, a round product into a square hole. It's got to work. And so I'm the project manager in Antipodean. And how this all started, well, once upon a time, we had a biochemist, Mike Murphy, who's that guy in the middle, and he was on sabbatical in Dunedin, and he linked up with Rob Smith, who's the bald guy in that one, and they were having a few beers, and Rob's a chemist, and they came up with this idea. What they wanted to do was to target antioxidants into mitochondria, and so they used coenzyme Q, and they used a targeting compound. So they had a phosphonyl and they surrounded it with benzene rings to sort of hide it and to give it felicity, and then joined the two moieties with 10 carbon. They looked at between 3 and 15 carbons, but 10 carbon was the one that gave them the biological activity. And what they used was the positive charge meant that they could use the membrane potential across cells and into mitochondria to concentrate what was attached to it, which in this case was an antioxidant, and take it into mitochondria. And then they showed that it was effective. They used well, quite a few models, but one of the ones they used was Friedrich's ataxia skin fibroblasts, which are a lot more sensitive to oxidative damage. You can see that with MitoQ, which is the blue, it protected the skin fibroblasts at a much lower concentration than the untargeted CoQ or its analogue idebenone. And that was all done at the University of Otago, and at that stage they'd sort of run out of, didn't have the funding or the expertise to develop it into a product. So Antipodean was formed, and in 2001 it had local investors, and also Force came to the party, and we started to carry on development of the product. It was basically still preclinical, and we ran it out of Auckland. And in 2005 we ran out of money, as you do, and we did a Series A, and that was basically US investors. So it's just over 50% New Zealand owned at the moment. And because we had US investors, we had to set up an office in the US. We've now completed 2 phase 2 clinical studies, and we need more money, but that's another story. Whoops, I did 2. Which one? Okay. So where I came in was at about this stage we were planning for phase 1, and so I just thought I'd just say some of the challenges that we found, that things don't fit on that pathway that you expect. And also Antipodean is a very small company. Basically we contract out all our services. So what you need to think about is, as I'm talking through the different people we work with, most of the expertise that has led to the development of MitoQ has been in those companies, not necessarily in Antipodean itself. So we had to work with Rob Smith at Otago and IRL to actually upscale volumes from batch scale to kilograms. Obviously the product has to be stable. We found we had to have a mesylate salt, and complex would be a cyclodextrin. We worked with Douglas because we needed to actually make tablets, and they needed to be in acceptable dose form. It needed to be stable. It needed film coating to protect it. And then the preclinical program had to be done in advance, so you knew what your ADME was, you knew your toxicity before you could actually give anything to man, which I'm sure you guys know that. And then working with CCST in Christchurch, and we did a standard SAD and MAD study. Before we started it, we used allometric modelling to try and predict what we should use for the first dose. I mean, that's always the hardest thing. The first time you give a product into man, how much do you give? We knew the plasma concentration we wanted to achieve, to give protection in mitochondria, and so we used, upscale from rats and dogs. And we gave the first dose to man, and we got exactly what we didn't expect. We got a much higher bioavailability than we expected. So you can do all the preparation you can, but you're not actually going to always get what you think. Phase 2, again, you're upscaling your manufacturing. Again, at IRL, you're producing your tablets. So when you get to phase 2, you've got to make sure that your studies are really robust and blinded. So one of the things we had to think about was MitoQ isn't white, it's yellow. And so when you're doing a placebo, your standard white placebo tablet's not going to work, because should a patient decide to have a look inside, they're going to be unblinded. So that was more of a practical point of view. And from a manufacturing point of view, when you're upscaling, sometimes things go a bit wonky. You find you've got new impurities, or your stability isn't the same, and you need an awful lot of product to do 9-month 9-month tox studies in dogs. From a clinical point of view, using an antioxidant, there are an enormous number of indications we could study, so we had to be very careful trying to pick which indications had a good rationale for the product, and also where there was some prior art, there was something similar had been done before. And also the feasibility of doing things locally. So we've done 2 phase 2 studies, one in liver disease and one in Parkinson's. They were the standard randomised, double-blind, placebo-controlled, 2 doses in studies, efficacy and safety. We had 30 patients who had liver inflammation. They had Hep C, which was either unresponsive or they refused therapy. And so we were looking to see if MitoQ could reduce the liver inflammation as measured with ALT. And that was a pretty standard study, pretty typical for the industry, not that hard to do. We did another study in Parkinson's disease that was more difficult from the implementation point of view. We actually got 128 patients in 5 months, which is 40% of all the people diagnosed with Parkinson's disease in New Zealand at that time. And that's huge. That's the kind of recruitment rate that you dream of and hardly ever get. And we achieved that by having an active recruitment strategy, really promoting that it's a New Zealand drug, it's homegrown. We're on TV, NZ, TV3, everywhere, the front page of the Listener, and that was really well executed. We're now looking at phase 3. Again, we had to upscale the manufacturing, and we've got to the point that actually we can't do that in New Zealand anymore. We had to go to a US manufacturer because we wanted to make 100 kilo sort of batches at a time. We're still looking at using Douglas to